Okay. I hope you can all hear me okay. Amanda, can you hear me when I'm
Yeah.
okay, great. So thank you everyone for joining us today for October Funk Lecture. My name is Marta Jarzyna, and I'm the Vice President for Public Affairs and Communications at the International Biogeography Society. You have seen me here already in the past, so you're hopefully familiar with me by now. But if not, as I said, I'm the Vice President for Public Affairs. So I deal with anything that has to do with the Funk lecture series, with communications with the society, and you can email me if you have any questions about anything that I've mentioned already. But today I am very excited that we'll have Dr. Amanda Bates to give a Funk lecture. Before I introduce Dr. Bates and before we start with the lecture, I have a couple of announcements. Let's see. Um, we have a series of workshops coming up through the IBS. The next one is coming up in a couple of weeks in late November on species distribution modeling for conservation using Wallace. And then we have a couple more workshops scheduled for early next year in February and then in April. So please check them out. They're listed on our website, biogeography.org. You can sign up through the website as well. Members of the society will have to pay $65 per workshop. If you're a non-member, it's a little more expensive. So if you're interested in taking these workshops, please consider becoming a member because it's a little bit cheaper when you do that. And then you get other perks as well with that. You're probably familiar with our biennial conference. The next one is coming up in January in Aarhus in Denmark in 2026. So not this coming January, but the year from now. So keep that in mind. We'll probably open abstract submissions sometime in late summer or early fall of 25. And if you have any questions or comments, we'll be taking questions at the end of the lecture. Please feel free to put any of the questions in the Q&A box, and I will read them out for Dr. Bates at the end of the lecture. You're welcome to raise your hand and ask a question as well. So it's kind of up to you how you choose to do that. Um, so I think this is all of the announcements that I have for today. As always, this lecture recording will be available on our YouTube channel after the lecture. So if you cannot stay for the whole time, or if you know of someone who would be interested in this topic but cannot make it today, feel free to share that information with them after the lecture. But now let me introduce our speaker, Dr. Amanda Bates. Dr. Bates is a professor and impact chair in ocean ecosystem change and conservation at the University of Victoria in Canada, where she also leads the Ocean Conservation Lab. Dr. Bates earned her PhD also from University of Victoria in 2006, and then subsequently she held multiple research positions around the world, including at Deakin University, at the University of Tasmania, and at the University of Southampton, before she returned to the University of Victoria. Her research broadly focuses on understanding ecosystem resilience and functioning, especially in the face of anthropogenic change. She aims to develop a process-based understanding of the factors that create species and community resilience by linking organisms' physiological thresholds to their environmental experiences. She then uses this knowledge to quantify changes in species distributions and the resulting changes in community dynamics. Recently, she volunteered her expertise for the inaugural biodiversity climate change assessment that was commissioned by the United States Geological Survey here in the US. So this is what we will hear about today. So without further delay, I turn it over to Dr. Bates. Let me stop sharing. And then Amanda, please you're free to share your screen.
How's that?
Looks great. Sounds great.
Great. Okay, so welcome everyone. Thank you so much for taking an hour out of your day. I was delighted to be invited as a to give a Funk seminar. I like of course, I'm a huge fan of IBS, and initially I thought I would talk about my research, but I was recently invited to participate in an assessment, a rapid assessment. So this was very short, three real core working months to do a rapid assessment of biodiversity change in North America. At the time, how much or how important studies of biogeography are in terms of producing evidence for whether or not we are seeing systematic change in natural systems. And so, first, I really want to thank IBS and the seminars series for inviting me. Thank you so much. And I guess I also really want to acknowledge and thank the community, because what's interesting is that as you start to pull apart evidence for biodiversity change, what I really see is that so much creativity and thought and really challenging analysis kind of emerge as we've started to document, evidence for biodiversity change and climate, as well as some controversy. And the USGS commissioned in 2022 a assessment for North America. And for me, I came aboard with this assessment because the US decided to include Canada and Mexico in this assessment, acknowledging the transboundary components of North America and the potential for biodiversity change in the US to impact both Canada and North America. So this is a 14 chapter assessment. We met last May and had a kind of inaugural meeting where we all shared ideas and started our chapters. And our draft was due on September the 23rd. And that draft is presently out for review. And I'll be talking about that today. And I also want to send a shout out. I had the macroecology lab in Southampton where I had teams working on biogeographical questions across marine and terrestrial systems. So a shout out to those who are from my Southampton lab here today. And then also I worked in Canada in a very cold place, Newfoundland, and was very interested in winter climate change. And these kind of perspectives and backgrounds really shape how you, I guess, approach, this kind of thing like an assessment in terms of what you may pick out from, a 30,000 page document. like it's sorry, 30,000 words, sorry, not page, 30,000 word document. So it's quite a lot packed in, but in terms of where I find the most interesting components. So I've just popped up the website here, and the material that I'm presenting today is all in draft review form. And we were literally working on it right up until the last minute, as you do. And we now have 2 or 3 months to take a step back, carefully think about what we've done, and we very much welcome your feedback. So you can, if anyone's really interested, please look at the full document here, and you're welcome to comment. So I'd like to shout out the co-chairs for this assessment, this 14 chapter assessment, as well as to the guidance committee that have really given some thoughtful and careful perspectives, for instance, such as how do we include Indigenous perspectives within our chapter, for instance. So our guidance committee have gone through all of our drafts for all of the chapters, donated their time, and it's just a huge pro bono effort. It's quite impressive. And what I'd like to do today is talk a little bit about chapter 3. We had a large co-author team, and for our chapter, we were given 30,000 words. And so when you get something like this, okay, you're going to do this rapid assessment of North America. My coordinating lead author, Catherine Suding, and I from the University of Colorado just kind of sat down and thought, how are we going to approach this? What is our, effort going to be? Are we going to produce some sort of glossy type, summary? Do we want to dig into the evidence? Do we have time to do a meta-analysis? What components of biodiversity change do we want to look at? Et cetera, et cetera. And we ended up engaging a lead author team across largely the US and Mexico from our lead author teams that, were looking and had special kind of interests from genetic diversity to, say, regional scale change and extinction. And then we have a huge list of contributing authors that have supported us in so many ways, from helping with literature reviews to doing figures and designs, as well as helping even at the very last minute when we were trying to get this in for this deadline, references, etc. So huge team, and I'll just give a shout out to so many who have made this like actually a really fun experience, which I wasn't expecting. I just got to say. But anyway, it's been really fun. Okay, so biodiversity change. We had to start with what's biodiversity change? How are we going to look at this, and what is the lens that we're going to look at this through? So when we were started off, we were asked to use the IPCC and, of course, the IPBES frameworks for how we define climate as well as how we define biodiversity. And so the charge, it's really okay to look at biodiversity across a whole bunch of different scales: genetic, phenotypic, phylogenetic, these functional aspects in time and space within species, among communities, and then also potentially tied to function and services. Although within the assessment, there is an entire chapter dedicated to function and services and aspects that are important for human well-being. So in our assessment, we focused kind of more on these lower levels, from genes to species. And we took a very broad view, and we acknowledged that like our concepts of biodiversity are firmly rooted in a Western science framework. So when we were thinking about biodiversity, and what was pointed out in our assessment meeting is that we're collapsing biodiversity down into kind of quantitative measures. And I pull an example here from my PhD candidate's work, Karen Richard, who's worked in a co-knowledge production project in Nunatsiavut with Inuit. And Karen's looked at populations of seabird change across, chunks of North America, focusing on Nunatsiavut, which is has hosts about 30,000 Inuit. And what we rapidly learned is that, okay, we're looking at change. We're interested in abundance changes going up or abundance changes going down. But our colleagues who we are working with and have been hired on the project to work with us and do the research have a different kind of conception of biodiversity change. And this is Jacqueline Winter's daughter. She's holding an eider duck egg. And for Jacqueline and many of her community members, biodiversity change is about sustainability in that cultural aspect, which is to be able to harvest these eggs into the future for both her and future generations. And so we're acknowledging, I just want to acknowledge up front, and what we were asked to do from our steering committee is to make sure that we were being direct, and I guess acknowledge where our perspectives come from. And so with that, focusing primarily on this Western perspective, when we look at biodiversity change, what we see in North America is we can really quantify patches of biodiversity change that cross different scales. And this is an IPCC generated figure, which just looks at historical temperature change, where the really, really dark red is lots of change. And then we've got the potential for cooling in the bluer, kind of cooler colors. And of course, what we see is across North America, just with temperature alone, we're getting these real differences in temperature change where, for instance, in our polar or Arctic spaces, we're seeing the most rapid changes. And if we think about biodiversity, then this task of trying to tease out how biodiversity is changing given different scales of space and time is obviously quite challenging. And when we, in our chapter, thought about climate change drivers, we were given the task of direct climate change drivers. So we focused on including studies that looked at CO2 forcing. So for instance, you could have a direct impact of CO2 through photosynthesis. CO2 is influencing heat content on the Earth, which is in turn influencing a number of other climate drivers as well as precipitation. And then, of course, CO2, which is being absorbed into our oceans, is influencing the pH of the ocean. So we have all of these factors that combine that can then accumulate to change biodiversity. And there's a suite of cascading drivers. And so in our chapter, we tried to be as inclusive of all of these drivers as we could, thinking about dissolved oxygen, salinity, et cetera, storm, hurricanes, ice cover. And of course, um, especially in the last decade, there's been a real focus on extreme events in the literature. So it's not just this kind of gradual forcing, but events that really push systems. And we see this kind of rapid response because of increases in variability. And so in this panel on the bottom C, what I'm just showing is a change in mean. You can have a change in timing of temperature or a change in variability in temperature, so greater extremes. And all of these factors, we wanted to be inclusive of all of these factors when we started thinking about how biodiversity is changing. And of course, this is IBS, we all know this, but the first challenge is, okay, scale. Let's try and be explicit about what scale that we're thinking about. And what I'm going to take you through today are the responses that we looked at local scales and regional scales, and how we've drafted this up to think about this as we can have genetic diversity change, for instance, being inclusive of that best definition at local or regional scales. And then we can think about the mechanisms that might be driving biological change. So we could see population change. We might see changes in the timing of life cycle events. And that timing in the life cycle events might then in turn change populations, or it might shift species interactions. And then all of these factors can potentially combine to drive patterns in species diversity. And then we thought about range shifts and food webs and how these might then transform ecosystems, where ecosystem transformation, and certainly from the paleo literature, is what was reported across since the Holocene in North America. So we really thought about this biome transformation or ecosystem transformation. So this is the approach we took, acknowledging that there were other kind of approaches that we could take. And we decided that even though we had 3 months, we were going to try as hard as we could to do a standardized review. And we wanted to do this review for all of taxa and systems in North America. And of course, what we wanted to do is just pull off papers where there was a really strong link or attribution to climate change in the study. But this is very rare. And so we ended up using a generous attribution criteria just because of this rarity of studies. And what we defaulted to was the interpretation of the authors. And we, at least in most of the work, didn't consider forecasting and modeling as direct evidence. So we were looking at studies that had, directly observed a response rather than forecasted or predicted a response for this chapter. And we've pulled off recent syntheses. That was our golden kind of standard. We were hoping, because of how quickly we had to do this, we're going to pull off recent syntheses and kind of look through those recent syntheses, supplement that from the literature and look in the literature, and then pull in any relevant literature as we drew in experts to kind of critique and draft our chapters. And we also tried to find this observational data, which were focused on a specific place where we could at least try and link a mechanism to change. And then we also tried to find long-term data. We also used experimental work when that work kind of supported and supplemented what we had found. Okay, so what I'm going to do today, and I'll just acknowledge this is the first time I've talked about this, so I may end up skipping a couple of sections if we run out of time. But what I'd like to do now is just walk you through each section. And what I'm going to do is just give you a little vignette of some of the methods, some of the results, and the main conclusions that fell out from each of these. In addition, I also led some reviews on Indigenous knowledge, as well as the paleo literature, at least the recent from the Holocene, the last 10,000 years, as well as species distribution models and urban biodiversity change. If anyone has any questions about that, please ask, but I don't think I'm gonna have time to go into it. I've got the slides at the end. But so what I'll do is just really focus on what our lead authors did in this team for these components, acknowledging that there's lots more detail here and some really interesting work. So hopefully it will come out in publications as we move forward with the assessment. Okay, so let's start with genetic diversity. We knew we weren't going to find a lot of studies that documented a clear relationship between genetic diversity and climate change. And when we started, we did our review, we returned about 400 papers, and very few of them were actually had direct links. And when we started, what this image shows is a little chipmunk and also a stonefly. Both of these are cold adapted species. The authors knew about these examples, they're kind of case examples, and we had a really hard time moving from these case examples that were already well known in the literature. And so here's a lovely little example of a chipmunk on the left, and it's moving up and up on a mountain. And there's been a shift in the effective population size, the number of individuals in the population that are going to return genes to the next population. So the effective population size in this case has been reduced. So a reduction in genetic diversity and the effective population size in this chipmunk as its range has been decreased on this mountain. In addition, there's a little example of a stonefly, and it has decreased connectivity. It's leading to lower gene flow. And so some of these case examples continue where we have cold adapted species, which we can see are undergoing genetic diversity change. So the authors in this chapter focus then on adaptive responses to climate change. And again, concluded that these adaptive responses are rare. So there's not a lot of evidence of this for North America in the literature. And this is probably related to the challenges of doing this and clearly demonstrating that mechanism through time, as well as attribution. But there is some evidence that's emerging, and that's especially for species that, of course, have short generation times. And so an example was a field mustard, which adapted to drought in Southern California with quite strong support. Invasive, Drosophila, I swear, I've seen Drosophila adapt to me vacuuming them up and become faster. So they escape my vacuum hose after a month and know I'm coming. So I feel Drosophila, if we're not going to see it, Drosophila, what are we going to see it in? So Drosophila has shifted its chromosome inversion frequencies in warmer temperatures in North America. There's also an example of a salmon, a cold adapted salmon, that is migrating earlier in a 3-decade period of warming, and that's been linked to genetic change. So there are some examples of adaptive change. So why aren't we seeing it more though? And this was, I guess, what was really interesting coming out. Is this because of our inability to measure and attribute? Or could it potentially do because there is quite a bit of plasticity, non-genetic plasticity that is coming into play? Or maybe it was that genetic change was not possible. And so certainly we expect change, such as, for instance, the salamander species. It's observed across a cline. It has a really strong heat tolerance relationship across a temperature gradient. But, in a series which looked at, change in this species over 40 years, they did not see any adaptation in heat tolerance in this 40-year period. So even when we would expect some of that change, we're not seeing it. And I think this really calls for studies that are trying to link genetic change with changes in traits and population level change. So we see in where we actually do have evidence, it's quite mixed. And of course, in some cases, the authors have pulled out some examples of changes in genetic variation or diversity or adaptation, and the implications of that can vary, especially for adaptation to change. So overall, we didn't see a lot of strong evidence for shifts in genetic diversity in spite of expecting this to be the case, and especially not at regional scales. So a lot of our studies were at local scale that were available. Okay, so that's genetic diversity, perhaps no surprise. So what I'd like to do now is jump to population change, and then I'll go through these other mechanisms of timing and species interaction. And so certainly going into the population change component or section of our chapter, we did expect there to be overall net population losses, especially in areas that were warming. And this is certainly informed, say, for instance, a study looked at the IUCN species which have been assessed. And so these are largely vertebrates. You can see images of the vertebrates on the y-axis here, as well as, insects. And many of those species are declining or stable at best. Many are unknown as well, but only a few or a handful, say looking across all of these, thousands of species, are actually increasing. And so Racine, the lead author of this paper, did a literature review. She pulled off about 500 species, 160 were left that she could actually use. When she looked at the mechanisms that were attributed to climate or climate drivers that were attributed to change include temperature, precipitation increases or decreases, drought, acidification, salinity, dissolved oxygen in water, and then fire on land. And overall, mixed results. So she found evidence of decline, similarly, evidence of decreases, mixed response in any studies that were looking across many species. And in a handful of cases, there was no change observed, but change was absolutely the most common response. And so generally, Racine concluded that local population trends are highly variable, and I guess this would resonate for anyone that's looking at how communities are changing in time and space. They're highly variable, but we're still limited on, our large scale data sets that can do this. So I want to just a huge shout out to IBS and many of the authors that are here. It's your data that many of these assessments are using. It's so critical. In addition, we found that data sets that could incorporate short-term variability and responses to that short-term variability were extremely important in the attribution kind of frameworks, because we can detect change and then we can attribute a particular driver to that change because of the short nature of it. Now, whether or not that short-term event is due to climate, of course, is debatable, but we know that these short-term events are increasing in their frequency. And then Racine also found that linking demographic traits to population abundance and persistence is really, really challenging. And that's probably no surprise. But I guess, again, it's these extreme events which can help us do that in a much more, I guess, defensible way. And what Racine also found is that oftentimes population or demographic responses can be buffered by behavioral and physiological adjustments. So again, we're starting to see kind of a trend coming out that potentially non-genetic change is really helping on the capacity and building kind of resilience or the ability to cope. And so Racine concludes, especially because of her expertise in the ocean, where we've got critical life history stages that are in the ocean, but she really found that any type of management intervention that is aiming to boost critical life stages and monitor changes in these key demographic rates, looking at both, is going to be so important in influencing and enhancing our understanding for how climate change is influencing all species, but very much so vulnerable species. So for those of you that are thinking of your next grant idea, I hope this is inspiring. Okay, so we've gone through genetic change and population change just briefly. So now I'm going to jump and vignette timing of life cycle events. And before we jump into that, I just want to highlight that changes in phenology have been one of the first responses, such as cited by Parmesan or Johan Parmesan, in the days when this was really raised as biodiversity is changing in response. But in our assessment, what our interest was is in seeing whether or not we were seeing positive or negative change. So we really wanted to try and link what we know is happening in terms of shifts in timing, which could be considered an adaptation to climate change, something, that's occurring, that's a shift to allow a population to cope. We wanted to see if we could tie that to population level change and specifically changes in demographic or vital rates. So is that driving that change? And so Nick Haydad looked at this with a team, and he relied heavily on a recent paper that was led by Amy Lur. And I don't know if I'm saying Amy's last name correctly. I haven't met her yet. But Nick looked at this paper, and he looked at 79 papers that took place in North America, which aimed to look at this link between population phenology and population change. And while there's certainly all of this evidence linking the climate change to effects on, demography, and we highlight the potential for strong consequences, we're still seeing a real lack of evidence for this. And in fact, Nick reports that he couldn't find any direct studies of phenological change on biodiversity, which is quite interesting. And most studies were also focusing on, say, plant flowering time and birds. Were really missing studies kind of more broadly, looking across many different and diverse taxa, which is true, I guess, up for all of the studies we've looked at up to this stage. And so we couldn't really tease apart, a real negative response in terms of how population phenology and changes in migrations or where species are reproducing. We couldn't necessarily link those things to a response in terms of a biodiversity impact. And thus, we've kind of concluded, wow, this is inconclusive. We're not sure if our phenological change is actually leading to biodiversity matches. And because I'm not going to include my a review on Indigenous knowledge here that I did with colleagues, I do want to flag that this was interesting because in the Indigenous communities across North America have been noting phenological change for decades and reporting it in relation to climate. And this phenological change can influence culture. It can change when harvesting, food is harvested. So there's a lot of implications for it that can be a loss for humans. So I want to point out that in our empirical evidence is inconclusive, but I think if we push a little more and think about how these changes are actually impacting humans, which are tied to place and space, shifts in phenological timings are actually going to, influence humans. So I'll just flag that here, and I won't talk about that in detail. But I think what's key in terms of just jumping back to some of the more biology of this is that there's mismatches in phenologies between independent species. And what we're seeing is that this can have advantages for some species and disadvantages for others. So overall, we're seeing this really mixed bag with winners and losers. And again, we need more research on this. We've got a real focus on plants. There was only 2 studies of the 79 in North America that addressed these population level consequences of phenological shifts. So even for plants that we know so much about, we're still got quite a knowledge gap. And so just like in the population change kind of section, Nick also really, focuses on, wow, we're really missing understanding of how demographic vital rates are really, really shifting. Okay, so now what I'd like to do is jump to species interactions. And species interactions was, of course, extremely interesting because we expect climate to potentially shift of species interactions. And I have to, oops, okay, it looks like I've put range shifts here. So we'll jump to range shifts and then species interactions. So in range shifts, we were just talking about phenological change and timings. This, of course, relates to range shifts where species can cope and move and adapt, now not at a local scale, but at a regional scale. And Cascade Sortie worked on this with a number of colleagues, and Cascade really focused on pulling out meta-analyses that had even been conducted at the global level, and then subsetting those to North America. And she supplemented this with a team of experts of papers and location-specific studies. And I'm just going to highlight this chapter was really rich. We have a lot of information on range shifts, and I'll just highlight a couple of take-homes from her chapter. So first, what we're looking at is an estimated shift in kilometers per decade. This is either on the left elevation, so you can have the trailing warm edge, and it's the direction that edge is going. or the leading cold edge and the direction it's going. With latitude, we can have our leading cold edge, which direction it's going, and our warm trailing edge, which direction it's going. And when Cascade looked at North America and subset from the global, kind of literature, what she found is that, North America is certainly supporting what we know at the global scale for the warm edges on mountains and on with latitude. So warm edges are moving up, and then our leading edge, the cool edge, at least across latitude, is also shifting. So it's moving to track warming as well. However, she didn't see this response, which is interesting, on mountains in North America. And so this cold edge, this leading edge on mountains, isn't moving up.
Well, it looks like we lost Dr. Bates. Perhaps we can give her a minute. Maybe she will be able to rejoin in a second. So let's just sit for a second and I can email her also and see if she is coming back. Just send her an email. Hopefully the connection on her end will come back and she'll be able to rejoin in a second. Hi, Amanda.
Oh, I'm so sorry, our power just completely cut out. So I had to wait for our internet to come back on and reset.
No
So I really
problem.
hope that doesn't happen again. Sorry, everyone. Are we okay? Can I keep going? I don't
Yeah, keep
even.
going. Absolutely, keep
You're
going.
okay. Keep going. Okay. We'll keep going. Carry on. So I think where I was when our power cut out here, which was really exciting, there was lots of beepings, um, is I was talking about the leading edge. And what Cascade found is that on mountains in North America, what's different than the global study is that this leading edge on mountains didn't shift upwards, which is what would be expected and predicted. So this is a little puzzling. And she digs into this in the chapter which I've given you a link to if you're interested in that specifically. So even though most range shifts are really to, track warming to what were historically cooler, again, what Cascade is seeing is that a lot of these, about 50% of species and assemblages are linked to warming in the direction that's expected. But again, are not carefully or not able, it's really challenging to attribute those responses directly to climate. But what we're generally finding is that when species do shift, it's faster in regions that are warming faster. And in general, more than half of the species are shifting in that direction, but half aren't. And so when we start adding other factors into our models, a great one on land is precipitation. What we see is that we get better explanatory power for many of these shifts. So temperature is not the only driver. Many other variables are important, and many of these other variables might be explaining some of the, I guess, unintuitive shifts that we're observing. And then what Cascade and her team also really highlight is there's many other factors at play, including habitat availability. So this is where we really have a chance to, intervene or implement conservation strategies that support species from their shifting. Okay, so what I'll do now is move to species interactions, and I was really excited by this section. So you can see I'm really keen to get to it because this was relatively new. There are no systematic reviews of species interaction, and the team that led our species interaction group, so this is Laura Pruitt at Washington, University of Washington, did just an awesome job. So I'm super excited to get to share all of her hard work. So Laura tackled this with a postdoc and looked at climate change, species interactions, and ecological impacts. So very kind of novel. She looked at climate change drivers, and what emerged from the literature as being the kind of the responses that have been most tightly linked to species interactions were wildfires, precipitation, changes in water temperature, and extreme events. So again, we're seeing this signal of extreme events as being where we have some power to attribute something that's related directly to climate to a response. And what Laura did with her team is look at competition, herbivory, predation, parasite, and diseases, as well as scavengers and mutualists. So these were kind of the species interactions that the literature really focuses on. And then in terms of ecological impacts, whether or not, some aspect of biodiversity was increasing or decreasing, abundance, did it increase or decrease? And then Laura also looked at ecosystem functioning. Are we seeing, increases in nutrient cycling, productivity, or stability in the system? Okay, so what I'd like to show here, and I haven't done this for all of the different sections just because of time, but when Laura and her team pulled off different ecosystems, you can see the diversity here, many tropical, desert, tundra, around to forests. Marine were clumped all together because of rarity in terms of, how we've looked at this, but will largely be, say, kelp forests or coral or sponges gardens. And then freshwater was also linked together. So a lot more detail on this coming out of terrestrial systems. And when we look at taxonomic groups, very few, for instance, bacteria were pulled out in this particular review, but we have much more evidence coming from our vertebrates again, a bit more from fungi, but plants again dominating this research. And then in terms of climate change type, temperature really pulls out as being the dominant driver here, as being one that we can measure and track through time easily. And so we're attributing change to temperature. And then the climate change impact was either a shift interaction, so it could flip the interaction. You could have a decrease, an increase, or no change. And the donut here, the slice of this donut pie, is the number of relative studies that were kind of pulled out for each of these. That's typical for these kind of diagrams. So you can again see the kind of, strong domination in literature of the terrestrial literature, and then of course, temperature. Okay, so what did Laura find in her team? Well, again, we've got a bar plot, number of studies on the x-axis and the type of species interaction on the X as being the ones that kind of emerged, and you could be able to kind of come up with a take home from these particular studies. And then the color here were the interaction changes, either a shift, a decrease, an increase in the interaction strength, or no change. And so interpreting this, what we see, for instance, is that competition, we see a flip in that relationship. With disease and parasitism in this kind of yellow color, we actually see an increase in the strength. And then for mutualism, no change or a shift, this can also occur. And in predation and herbivory, we see kind of a decrease in the strength of that predation or consumer. And this is kind of summarized then that we do see really strong kind of relationship between these different interaction types. So this work is really kind of highlighting the potential for climate change to influence, interaction strengths, and how this is going to play out at local and regional scales is quite interesting. And so we get this flip in some cases, say with the competition dynamics from winners to losers. And predation and herbivory are really affected in system-specific ways that make generalizations difficult. So there's not really a clear signal coming out there. But parasites and pathogens seem to be emerging, or there's a publication bias and we're just pointing out where diseases and pathogens are emerging, not sure. But certainly we're getting an increase in evidence for parasites and pathogens as becoming kind of more harmful. And then this very handful of studies on mutualistic interactions suggest that these are relatively resilient to climate impacts, which is interesting. Okay. The other kind of take home is that there's, of course, as with all of these components, some real gaps. And something that Laura and her team really highlighted is that altered snow and fire regimes are really neglected in some ways as a climate change driver in terms of how they influence um, interactions between species. And then I guess the second stage, if we think about interactions and we're going to that regional scale, what we'd love to do is to be able to think about how local scale interaction changes influence regional networks. And that, such a phenomenal idea to kind of advance that and look at how these kind of patchiness in space and time of climate drivers could then influence, how interactions sum at local scales across regions and in networks. But there are very few studies that are achieving such a goal, such an ambitious goal. Okay, so I guess given time now, because I want to leave time for questions, what I'm going to do is just walk through some of the changes in species diversity here, and then switch to some of the more novel aspects on ecosystem transformation. And so Celia Simmons and Brian McGill Celia looked at local scale changes in species diversity, and Brian McGill looked at regional scale changes in diversity. And for many of us that are here, we've kind of been embedded or have probably read about some of the controversial kind of evidence emerging from what's happening at local or regional scales with diversity. And what Celia found at local scales is that, species richness was going up, it was going down, it was quite mixed. There was a really strong signal of turnover. So lots of change, which corroborates the population level change. And then when Celia looked at functional diversity change, the evidence to date kind of supports the same trend that we're seeing in richness. Functional diversity can go up and down, and there's kind of various ways to think about how we measure functional diversity, which can also explain these kind of up and down changes. But overall, seeing a really strong signal of turnover and a really strong change in terms of the types of species that might be shifting, such as shifting to species that have warmer affinities or that are more generalist in their character. But again, we were surprised at the lack of actual evidence that we could include in this assessment with a strong climate attribution factor. And so skipping ahead to some of what Brian did, in brief, Brian looked across regions, Brian McGill looked across regions in North America. And again, what he found is that spatial differentiation, so beta diversity, how different communities are in space, there was either in the middle, this is the number of studies, and Brian only found a handful of studies that, met our criteria. The green here is direct attribution. And what Brian found is that there were very mixed trends. So across, communities or no trends, or if we look towards the homogenization, so beta diversity is decreasing or species are becoming more similar, there was pretty balanced in terms of a very handful of studies that he could find that were with beta diversity is increasing or species are becoming, more spatially differentiated. So overall, we're seeing a mixed signal again. And Brian also dug into the regional and global extinction literature and especially looked at some case studies where we've attributed an extinction event in North America to climate change. And in every case, what he found is that, of course, the media, kind of highlighted climate change extinctions, but these climate change extinctions were often related to many other factors. So, for instance, there was a climate change caused mammal extinction that was attributed directly to sea level rise, but that species was in decline for a century already. Um, and, there was a cactus, for instance, that's been attributed to increase in sea level rise, but it's extirpated. It's actually established in many other Caribbean islands. So we're certainly seeing a loss of species and extirpation, and in many cases, biodiversity loss. But again, we're missing that kind of key evidence, or what we're finding is that climate change is an exacerbating kind of effect. And the chapter that follows ours, we're chapter 3, chapter 4 is tackling those interactive effects, which we're really supporting. So we're really seeing across our chapter that climate change is like a compounding factor that's interacting with many other variables to lead to population decline. Okay, so I think given the time, what I'll do is focus a little bit more on our ecosystem transformation and give you a bit of a background for this section because I think it's one of the surprising and novel aspects that's emerged from this assessment. And Katie Seding led this. So when Katie and I sat down, we kind of thought like, what are going to be the big transformations that happen? Katie works on this in part in her research program. And in the ocean, I work on coral declines as well as kelp declines and how that influences biodiversity. So first off, there is no common language to think about ecosystem transformation in marine, freshwater, and terrestrial systems. So that was interesting in and of itself. Ecosystem and transformation is used by the USGS to think about something like a biome shift. So what Katie did is think about all of the different kind of ecosystem transformations that you can have in marine, freshwater, and terrestrial systems. And she thought about like, what is an transformation and how could we score an ecosystem transformation response? And so in the top panel A here, we have stability kind of through time. On the Y, we have a biome at time 1 and a biome at time 2, but really you can think about this as, an ecosystem at time 1 and time 2 if you're not used to thinking about this in terms of biome, for instance, in the ocean or in freshwater systems. And then in B, there's some sort of event. So there's, a pulse of a disturbance. And in the case of B, that system recovers and the trajectory in the blue kind of falls. You see this, demise of that system. It's damaged, and then it returns to kind of a restored system. In C, there's kind of a slow press where you've got this kind of steady decline of a system transitioning from one biome to the other. And then in D, and this is what was really interesting in Katie's work, is that when there's an event, what we find is that the recovery trajectory, say there's some sort of disturbance event that's due to hypoxia in the ocean, a fire on land, or an extreme event, or even an event that's unrelated to a climate change driver, but something that damages that initial biome or system. So you have that event, what we're seeing is a failure to recover to whatever that system was historically. And when we dug into this and really thought about this, Katie realized that, this could be because that system was, say there was a lag. And so that biome at time 1 looked, healthy, it was functioning, it might be, full of species, but there was kind of a climate debt. And so maybe we could conceive that species the community didn't recover because of that climate debt or lag. In other cases, maybe that system is warmed. So the new species coming in thrive and outcompete the species that were there before. And there's a multitude of reasons for this. And what Katie did is spent a ton of time looking at the evidence across a whole bunch of different ecosystems on the left, from forests to reefs, kelp forests, as well as lakes. And then she looked for evidence of climate change kind of press events where there's kind of a slow change in variables, say from temperature or precipitation and sea level rise. And she found evidence of this in any of these colored boxes, which is the key point to look at this. How much evidence do we have for these climate change events that are just kind of a slow event? And then she looked at pulses and, of course, found severe fires or heat waves or hypoxia, which we expected and predicted coming in. We see these across many of these examples across these different ecosystems. And then she also looked at these kind of other drivers as exacerbating or influencing this event and found evidence in these colored boxes for this in many systems, such as grasslands and especially coral reefs, where other drivers are coming in and influencing these events in terms of transitioning between what we would say in marine systems states, ecosystem states. And then she looked at the mechanisms. So for instance, rain shifts or climate-related kind of trigger, climate disequilibrium, for instance, where we've got this, the community being out of equilibrium, and found evidence for this across the board for these very different mechanisms explaining these failure for one system to transition to the other. And then she also looked at resilience. So where are we seeing resilience when we see it? And what was the mechanism that was actually conferred there? And so this could be, for instance, response diversity or network effects. So that was quite interesting. And across marine, and terrestrial systems, we're seeing this kind of multiple kind of forms of evidence in the resilience in the system. Okay. So I'll just check in on time, and I've got about 3 minutes. So what I'd like to do then is just jump to some conclusions. What are the main takeaways as we move across North America and think about this huge effort, this huge assessment? Where did we come to? And what we've done here is in this slide, the orange are terms that our USGS assessment asks us to use. They go from, say, unresolved to established but incomplete to well-established. So you can look at that level of kind of confidence in the quality of the evidence. And so overall, we find really sparse evidence for changes in genetic diversity and adaptation. Most of the studies that have investigated genetic change have documented diversity change in cold-adapted species, and population growth and decline trends are inconsistent across regions. And on shorter timescales, we are really seeing strong responses to mass mortality that's reshaping biodiversity. And, what we're also seeing is that we've got some climate change winners and some that's relatively, kind of unresolved in the literature, but we're seeing losers. So species that are cold adapted are definitely losing kind of across all of these different studies that we've looked at. Climate change is certainly influencing the timing of life cycle events, but how this scales outwards to other aspects of the community is really inclusive. And climate is affecting a huge broad range of species interactions, but how this is then translating on these interactions and how this is influencing populations is less well known. And indeed, as we move towards food web, we're starting to establish that there's food web effects, but the mechanisms and the scales and the context dependency are quite incomplete. We know that geographic range shifts are certainly occurring, yet the rates and responses and how they're occurring is very variable, and that's very well supported by our assessment. And then we have fewer shifts that have been documented than expected based on projections. And so we're really curious, why aren't we seeing the level of response that we would expect based on projections? And so we have overall minimal evidence as well for species extinctions, even though turnover is really, really high. We have significant shifts in functional diversity in many, many taxa that occurs in some cases with shifts in taxonomic diversity, but we don't know if this is going to bring ecosystem function. We've also got limited estimate for regional homogenization of diversity, which would be expected if we're getting winners of the same species, but this is so far, at least in North America, inconclusive based on the papers we were able to find. And we are seeing this widespread pattern of ecosystem transformations across all of our systems, which is also predicted from the paleo literature. And so we are seeing change, but interpreting that change and the lens on which we interpreting that change is really key. And what we really push for is kind of the final closing points is that observations for detection and attribution are so needed. We are seeing evidence for a lot of adaptive capacity in nature, especially when species can move, can cope, and have that non-genetic, capacity to cope or adjust, and when they can move to new locations. And when we look back in the paleo literature, we see that this is absolutely critical for preventing, extinction. So allowing species to be able to move is going to be so key. And then we also are seeing evidence really coming out that climate refugia are harboring unexpected levels of biodiversity, including genetic variation, and that this is a really key area for future investigation. This is supported by the paleo literature as well, but we're also really seeing this come out in terms of the collective studies that we pulled off here. Okay, and I'm sorry, I think I went over a couple of minutes. I'm sorry about that. And this was the first time I gave this talk, so I wasn't quite sure how it was going to go. I think I had too much, but I'll leave it there and just flag to everyone that you can absolutely go and put comments and look through and just this draft that's out for review now. Thank you.
Thank you so much, Amanda. I will stop the recording now.